Okay, it's my pleasure to start off by introducing Holly Martin tonight, who is here on the staff of Gillette College. And what we're going to do with our talks tonight is we're going to begin small and work large. And Holly starts us off with the smallest things, bacteria, which she studies in the context of something slightly bigger, the human body. Ms. Martin is an example of the excellent researchers and teachers here at Gillette College. Her area is biology. Here she teaches life sciences, microbiology, is in charge of the laboratories, and it's an important part of the general science program here. It's an important part of the nursing program that takes place here, and the Gillette College tells me repeatedly that they are very proud of. Holly's own research career began as an undergraduate at Morningside College in Iowa. Then she moved to not quite a UW rival, University of Nevada at Las Vegas, where she got a master's of science and then stayed to work as managing a microbiology lab. Now, laboratory science, as many of you know, is almost always cooperative, and during her time there, the team she worked with published 6 articles with Holly as the lead on either the lead or the second author on all of them. And I'd read you a couple of titles, but to be honest, I'm from religious studies. I can't pronounce the words in most of the titles. This summer, she won an early career faculty travel award for the American Society of Microbiology, and she went back to UNLV for more lab work there. And here, just to make one last comment, here at Gillette College, her research work includes supervising 3 students who are working part of the INBRE project, which is a multi-million-dollar statewide, what would you call it, applied research project that's run out of UW at Casper. Lots of cooperation going on here. And tonight she's going to talk to us about the biological arms race. Holly.
Thank you. So I just want to give you a general overview about how the talk's going to go. First, we're going to talk about, even though we can't see these bacteria, they are everywhere. Then we're going to talk about your immune system and how your body is going to be able to adapt and deal with these ever-present microbes. Finally, we're going to learn that not only do those bacteria sometimes evade our immune system, but they also have mechanisms to fight back. We'll talk about some of our ways that then we fight back against those microbes. And finally, I'm going to kind of sum it up with a little bit about how we research these antibiotic microbes. So like I mentioned, bacteria is everywhere. Not a surface on this earth have we not found bacteria present. We're constantly amazed at the new places that we can go and find more bacteria. So one you guys are probably familiar with is that bacteria are found in hot springs. And we have hot springs around here. They're fairly hot. But we found bacteria living in hot springs that are above 100 degrees Celsius. So this is hotter than boiling water. So they can survive in these conditions that we're used to thinking life can't handle. If we go to the other extreme, bacteria can survive in the coldest of environments. If you dig way deep down in the glaciers in Antarctica, there are living bacteria present there. So, all right, they're hot, they're cold. We can also dig deep into the earth and find bacteria present there. They've dug miles below the surface of the earth, and they have found living bacteria present way below things that we would ever expect there to be life. So they're hot, they can live where it's cold, we can't even go down and escape from them. Well, maybe we can go up into the sky and find a place that bacteria aren't present. Well, that's not true. We have studied, or people have studied clouds, and they can look in the clouds and they find active bacteria living in the clouds. So pretty much every place you could think on this earth, we have found bacteria. Now, you're like, all right, it's everywhere else out there. Maybe my body's safe. You're wrong. Not even close. There's actually probably 10 times as many bacterial cells in or on your body than there are human cells. So, kind of begs the question, what are we? Are we bacteria? Are we human? Are we some sort of hybrid? That's a talk for a different day, but it's something to ponder. Now, a lot of people have a very negative connotation with bacteria. You think bacteria, a lot of people think bad. Well, it's not really true. About 99% of the bacteria in the world is either neutral, has no effect on us, or actually beneficial. We need them to survive. There's a very small fraction of bacteria that do harm, that cause disease. So how are we able to deal with these bacteria that are constantly kind of bombarding us? Well, we have our immune system to thank for that. It is probably one of the least thanked systems of our body that has a huge task to deal with constantly. Now, I don't want to get too morbid, but if you have ever experienced something that's recently dead, you can see how important the immune system is. Even a few hours after death, bacteria, fungus have taken over that specimen and started to use it as their home. So usually this immune system is doing a really good job at, keeping these harmful bacteria at bay. And I want to give you guys a little bit of a look into how the immune system works. So there are 3 different lines of defense of your immune system. The first 2 lines fall within the innate immune system, and the third line is the adaptive immune system. So the innate immune system makes up the first and second line of defense. There are 3 major parts to the first line of defense. The biggest one is your skin. So I kind of envision this as the wall. It's the barrier between you and the outside world. So it wants to keep all of your stuff in and keep everything else out. There's a few different ways that the skin's going to work to protect you from those invading microbes. The first way is your skin is extremely dry, and it is that way for a reason. Everything we know needs water to live. There's nothing that we found that doesn't need water. So your skin's dry so that there's no free water available for those microbes to use to live. If you think about places where microbes are associated with on your body, think of like your armpits or your feet, those are the places that end up being very moist and therefore can then have bacteria living there. Another factor that your skin uses to protect you is the fact that it's always kind of shedding. You're always losing the top layer of your skin. And this is done for a reason. That top layer is going to fall off. Bacteria cells are tiny, so they're going to be much smaller than the skin cell that it's on. So if the skin cell falls off, anything that's on top of the skin cell is also going to fall off. So it's constantly shedding, so then you're not having these microbes be able to kind of take up residency on your skin. Your skin also produces some chemical factors that allow it to protect you from those invading microbes. There's enzymes that it produces that basically break down bacteria. Part of the oil that your glands secrete is actually toxic to many bacteria and funguses. The second part of your first line of defense kind of moves inward. Anywhere that there's an opening in your body is covered with a mucous membrane. And that's for two reasons. One reason is we don't want those surfaces drying out. If they dry out, then they're probably going to crack, and that's going to allow bacteria in through that hole. Another reason is the mucus is a barrier between the cells and the environment. The bacteria or invading virus can then get caught in that mucus, and then your body can get it out. So we would cough and remove it or sneeze, and it's going to get that bacteria or virus out of your body. Now the third line of defense is what's called the normal microbiota. And this is a little bit of a controversy. The normal microbiota is all the bacteria that normally live in or on you and doesn't cause disease. Now the reason that it's controversial is you're not technically born with it. The rest of your innate immune system you're born with, it doesn't change, it stays with you for life. Normal microbiota is going to adapt through your life, it's going to change, and it's technically not there right when you're born. You are colonized fairly quickly after you're born, and it's going to take up roots. Because of its protective nature, I think it belongs in that first line of defense. So these bacteria that are present constantly there, they're going to work in a few different ways to protect you from those invading microbes. And anything that's a microbe that's invading, trying to cause disease, is also called a pathogen. So I might say pathogen, bacteria, those are used kind of interchangeably. So the normal microbiota is going to work in a few ways to protect you from those invading pathogens. One way it works is it's already taken up that space. If we think of our gut, that's where most of this microbiota is present. It's already there. It's already found the real estate and called it its home. So it's harder for something to come in and like push it out of the way and be like, no, I want to live here too. So that's one way it's going to work. Another way it works is these normal microbiota can produce toxic substances to the other bacteria. So a way to keep them from invading their home is to release a toxin. Not toxic to us, toxic to the other bacteria. And finally, these normal microbiota can protect us by changing the environment that they live in. They can change the environment by taking a resource and using it up. So if it's maybe somewhere in your colon, they can use up all the oxygen. So now that a bacteria that requires oxygen can't live there because it's all gone. Now, for some reason, your first line of defense fails you. It's all right. We have our second line of defense, and that's going to come up and help protect you. Now, there's 4 parts of the second line of defense. One part of the second line of defense are different responses. One response is inflammation. So a part of your body is damaged. What happens is that local region is going to respond by swelling. So with inflammation, we usually see 4 things. We see the swelling, we see redness, we see it's usually hot to the touch, and it's usually painful. The reason for this swelling can be a few different reasons. One of them is trying to dilute whatever's causing the injury. So we'll put a lot of fluid there. Hopefully we can flush out whatever is causing this damage. Now the second reason, if we can't do that, if there's for some reason we can't eliminating this injuring substances, we can contain it. So let's get all this swelling there and block it off from the rest of the body so it doesn't affect the rest of your systems. The third reason is that it can also migrate immune cells to that area to help treat and repair those tissues. Now, swelling is a local response. It's going to happen in that area that's damaged. Another line of defense is going to be a fever. And a fever is going to be a systemic response. The whole body will undergo that temperature change. So fever is considered anything, above whatever your normal temperature is at. Now, the exact reason that your body goes into this fever state is not really understood. There's a few different hypotheses on why it happens. One is reactions occur faster at higher temperature. So when you are infected, you need to produce more immune cells to deal with this infection. So if your body raises the temperature, you can produce these cells more quickly. Another hypothesis is that maybe the microbes aren't able to handle that hotter temperature, so they won't be able to survive. And then some of the chemicals that are involved in the signaling within the immune system seem to work better at a higher temperature. So maybe that's the reason the body goes into the fever state. The third part of the second line of defense are what we call antimicrobial substances. So your body can actually produce things that it inhibits the growth of microbes. An example of this would be a molecule called transferon. Transferon is going to go around and bind all of the free iron in your body, which you might not think there's very much of, but there's some free iron. And a lot of these invading microbes need iron to live. So they're going to be fighting for the iron, and your body's going to try to store it away so the microbes can't get the iron. If the microbes can't get the iron, then they can't live. The fourth part of the second line of defense are a bunch of cells that are called phagocytes. Weird name, but what it means is they're able to ingest other cells. These are all derivatives of white blood cells, and they're going to go around, and they're basically scavengers in your body looking for anything that's foreign. If they find anything that's foreign, they're going to engulf them. So think of it as like a Pac-Man going around and eating things that's not supposed to be there. It kind of looks like a blob, and then it just kind of takes over what is invading. Do you have a question?
Yeah, what kind of foreign things are they invading?
It's the foreign bacteria or the foreign viruses.
She's not going to be able to sleep.
Now, we get to the third line of defense. So if our first line and second line of defense fail us, we have our third line of defense. This part of your immune system is referred to as the adaptive immune system. So with our innate immune system, it's present from birth, it doesn't change, it treats everything the same. This time, it's going to adapt, right there in the innate. It's going to learn, it's going to see microbes, learn them, and then tailor specific responses to specific bacteria. So hopefully as you age, it learns, and it can protect you. Now there's two parts or two branches of the adaptive immune system. The first branch is mediated by B cells. B cells are specialized white blood cells, and for once in science, we kind of named something easily for you guys to remember. They're called B cells because they mature in the bone marrow. So B and B. They are going to be involved in what's called humoral immunity. And what that means is if there's an infection in the body fluids, they're the ones that are tasked with dealing with it. So if there's infection in the blood, in your lymph, these guys are going to be the ones that go around and take care of it. The second branch of your adaptive immune system are T cells. Once again, we did a good job naming for once. T cells are white blood cells, special white blood cells. They mature in your thymus. So we have thymus, T cells. For once we can remember those. T cells are involved in cell-mediated immunity. So that means is if you get an infection inside of a cell. So viruses are going to infect inside cells, and some type of bacteria also infect inside these cells. The T cells are tasked with clearing and dealing with those types of infections. Now, both the B and T cells are going to work in conjunction with antibodies. And I'm going to use my favorite stuffed animal to help explain what an antibody is. So an antibody is a protein, and it's going to look like this. So Y-shaped protein, and it has two main parts up here. These parts are going to bind to what are called antigens. Antigens are found on the surface of cells. So we'll have antigens on the surface of these invading viruses, microbes, fungus, parasites. They're all going to have antigens. One antibody works for one antigen. So here we have a square antigen, and that's the only shape that's going to fit into this antibody. Now the B cells are going to have these antibodies on their surface. They're going to be on the surface of the B cell. It goes around and looks for invading antigens. If it finds it, it triggers the immune response. Now the T cell is going to work a little bit differently. Now remember, the T cell is going to mature in the thymus. One part of maturing in the thymus is undergoing a little boot camp. And during this boot camp, the T cell has to show that it can recognize self-antigens versus non-self antigens. So all of your cells also have antigens on their surface. And we do that so that your body knows what's you. So think of it as like a little flag saying this is home turf. This is our flag, we're waving it on our cells. The invading microbes are going to have another flag that's going to be on their surface wagging. And the T cells have to be able to tell what's our flag versus what is a different flag. People that have autoimmune diseases, a lot of times their T cells have a hard time recognizing what is a self-antigen versus a non-self-antigen, and they confuse self-antigens and then start attacking their own body. Okay. So hopefully, using these different branches of the immune system, your body is able to fight off these invading pathogens. Well, we have to remember that bacteria have been on this earth much longer than we have. They've had a lot of time to adapt, and they've done a good job of adapting to their different environments. We saw that at the beginning. They can live almost anywhere. They're good at adapting to these places. Well, they didn't stop adapting when humans came around. They have adapted strategies to deal with our strategies. So I'm just going to highlight a couple of them. Staph aureus, it can be engulfed by a phagocyte, and usually that's supposed to kill the bacteria. Well, once the Staph aureus gets engulfed by the phagocyte, it produces a toxin, and it kills the phagocyte and escapes, and nothing's harmed. Viruses such as HIV can prevent some of the crucial stages of what needs to happen once it's engulfed by that phagocyte to kill the invading virus. But don't worry, we don't just take this lying down. We have fought back as well. And one of the ways that we have fought back is through the use of chemotherapy. Now, usually people hear that and they think, okay, you're talking about cancer. But actually, chemotherapy is the use of any chemical substance to treat a disease. So taking antibiotics to clear your sinus infection is a type of chemotherapy. Now, in the late 1800s, the idea that germs cause disease was established. And this was given the name germ theory. Shortly after germ theory was established, a medical student named Paul Ehrlich decided, well, if it's caused by bacteria, maybe there's something that's a special bullet, this magic bullet, that we can use to put in the body and get rid of only that bacteria. Harm the bacteria, keep ourselves safe. Well, about 30 years later, Alexander Fleming discovered penicillin. There's some talk about him being a sloppy microbiologist. I would like to give him a little more credit than that, but we have to kind of thank him for this discovery of antibiotics. What I find interesting is the story of the first time that antibiotics were used. It happened in, I think, 1930. A policeman in London came into the hospital and he said he just nicked himself shaving, and all of a sudden his face was just swollen. He had an infection. It was caused by Staphylococcus aureus. The doctors did everything they could at the time. They ended up removing one of his eyes, trying to stop this infection. Well, one of the treating physicians knew about a research scientist who had been working on penicillin. And in the lab, they had about 3 milliliters of penicillin. And that is like a very small amount. It's like less than a teaspoon of an amount of liquid. The doctors are like, let's give it a shot. We're gonna lose this guy if we don't give it a shot. So the next day they gave him an injection of penicillin. Started to get better. Gave him a few more injections of penicillin, and he was remarkably better. Then they ran out of the penicillin. He started getting worse. So they started actually taking his urine and trying to isolate the penicillin out of his urine to give it back to him. Went for a little while, but ultimately they ran out of penicillin, and this man died as a result of their staph infection.
It's a hard story.
It teaches us 2 lessons. There's 2 very good lessons here. The first one, antibiotics work. The second lesson, you need a full course of antibiotics for them to work. So, if you ever get sick, please do me a favor and finish your entire course of antibiotics. That is one infuriating thing that happens when I hear about as a microbiologist, and hopefully this story will give you a reason why.
All 10 days.
So, one of the problems when penicillin first came out is people realized how effective it was. We lived in a world before this that there weren't antibiotics, and people usually ended up dying from bacterial infections. When it first became available, you could go to your drugstore and get penicillin over the counter. We soon realized that that wasn't very effective. People were taking them inappropriately, and not 6 years later did we see the first penicillin-resistant bacteria, and it was Staphylococcus aureus. So this summer I had the opportunity of going to the University of Nevada, Las Vegas to try to study these antibiotic-resistant bacteria and maybe come up with some strategies to treat them. give you a very quick insight into the work we did. One thing that needs to happen in all cells is the information within the DNA needs to be able to be used by the cell. And one way this happens is through a process called transcription. And there's machinery that go around and extract the information out of the DNA. Now sometimes this machinery gets stuck and it just sits there. It stops working and it just sits on the DNA and nothing will happen. Well, there's a protein called MFD that can come in and remove that machinery from the spot. After the machinery is moved from the spot, DNA repair proteins will come in and try to fix whatever caused that problem. Sometimes that DNA repair protein doesn't work exactly right and it makes a mistake, and this can lead to a mutation. These mutations can ultimately lead to new traits and new characteristics, one of those being antibiotic resistance. So I want to tell you a little bit about how geneticists do their work. When we're interested in something, what we do is we remove it from the cell. So I'm interested in MFD. I'm going to remove MFD from a cell and then see how that cell works. So this would be similar to if I was trying to figure out how a car works, I would step by step remove pieces from the car and then try to drive the car and see what would happen. So if I wanted to see what the steering wheel did, I build a car without a steering wheel and then try to drive it. So that's what we're going to do as geneticists. We're going to take parts out of the cell, see how the cell responds, to try to figure out how that part works. I need to tell you a little bit about how bacteria grow so you can understand this. When they're first introduced into a new environment, they're not going to grow at all. They need a little bit of time to get it introduced to that new environment. That's referred to as the lag phase. Once they're comfortable, they're going to take off and they're going to start growing. They're going to multiply rapidly and their growth is going to be exponential. And this is referred to as the log phase. Some point they're going to run out of nutrients, so their growth is going to stop, which is then going to be stationary phase. And either they're going to run out of nutrients, that place is going to be toxic, and ultimately there's the death phase. Now originally it was thought that cells could only get mutations when they were growing. Recent research has shown that cells can also get mutations when they're not growing. And that protein MFD plays a role here. Now, when the cell is growing and everything is fine, MFD leads to what we call faithful repair. It puts in the right base in that damaged location. When we get to stationary phase, for some reason, MFD switches and leads to unfaithful repair and causes mutations to happen at a faster rate. So what we looked at are two different strains. One strain with MFD, one strain without MFD, within Staph aureus to see if maybe MFD is responsible for some of these characteristics we're seeing. So the first thing we did was we looked at different antibiotics to see if MFD was important there. The higher the number, the more of that antibiotic it takes to kill the bacteria. So we don't see a difference with rifampicin, we don't see a difference with ciprofloxacin, we see a huge difference with tetracycline. We were not expecting this. We knew that this is actually a strain of MRSA, so we were expecting it to be resistant to things like methicillin, which is that M. We were not expecting it to see such an effect there at tetracycline. Now we're knowing that it's coming resistant to antibiotics at a really fast rate, so we wanted to see, does MFD play a role in this? So we tested how fast it's able to mutate with and without MFD to different antibiotics. We saw something really interesting. Depending on the antibiotic, determined if MFD was crucial. So rifampicin, it actually mutates faster without MFD. With tetracycline, it mutates faster with MFD. The next thing that we looked at was oxidative damage. And one of the things that happens after bacteria are engulfed by phagocytes is that they're treated with a bunch of oxygen radicals, and this is going to become toxic to the cell. So we wanted to see, does MFD play a role in protecting it from oxygen radicals? And it kind of looks like it does. The purple bar on the end would kind of suggest that it didn't, but when we looked at those cells a little more closely, we saw they were all clumping together, so that was probably providing protection there. The next thing we looked at was UV damage. UV damage is what happens when you get a sunburn. You're going to damage your DNA because of that UV light. This was going to be a measure of how well this bacteria would be able to adapt when they're put under pressure. And what we saw here is that MFD was essential for adapting when it was put under pressure. The final thing we looked at was if MFD played a role in what's called hemolytic activity. And this is one of the ways that the bacteria fights back against our immune system. Staph aureus has the ability to lyse open our red blood cells, one, for protection, and two, to get iron. What we saw was when MFD wasn't present, this ability to lyse the red blood cells was slowed down. So MFD was there making this process happen a lot faster. So it seems like MFD is involved in some of the aspects of disease progression in Staphylococcus aureus. Surprisingly, MFD was also involved in having to become resistant to tetracycline. Now this is my part where I don't want you guys to be freaked out and go home and start scrubbing every surface. It is a major area of concern. Antibiotic is a major area of concern. However, there are a lot of people working on developing new antibiotics and developing alternative ways to deal with antibiotic resistance. You've made it this far in your life, you're fine. So please don't go home and start freaking out over this. I just want to thank some different things. A lot of this research was funded through the INBRE, the Northern Wyoming Community College District has graciously allowed us to do this research. The summer research was funded through a program called REU. And then at the top are two of my research students who actually get in the lab and get their hands dirty with this. So thanks guys, and let me know if you have any questions.